So I'm not going to spend a lot of time on career path, actually, other than just to say what you see here, that you may think at this point that you have to go in a very linear direction, but rarely is that the case. And that's certainly not the case for how I ended up working in science. It's more like what you see on the right, where it's completely nonlinear. I went to undergraduate school and got a bachelor's degree in nursing at UAB. And then I went and I worked as a critical care nurse for quite a long time, which I have to say is a great thing to do. It is an exciting type of career. But it wasn't quite enough. I was very interested in understanding how it is that certain things happen, what was the physiology of it, what was the chemistry of what I was observing in the unit. And so I decided to go back to school. And as a nurse, you think, well, obviously, I'll go to medical school. And so that's what I had in mind. And so, like a lot of my friends, we started taking courses to finish out our requirements. And unlike a lot of my friends, rather than hurrying through the minimum requirements, I found that I really loved organic chemistry. I loved calculus completely. And so I took all the courses and took some more and finally realized that for me, basic science was probably the thing I really wanted to do. Although I would say I think medicine, again, is a fabulous career. So it's just whatever you really like. So as I was planning on going to graduate school at UAB, I got married. And the person I married took a job here at LSU. And so, fine, we moved here. And I went to graduate school here eventually after continuing to work. And when I went to graduate school, I wanted to do biochemistry. And I went and I did it in a plant biochemistry lab. So about as far away from mammalian work as you can get. But I learned a lot of basic things that apply no matter what you do. So when it came time to do a postdoc, because I had worked in the lab that, although it was plant biochemistry, we studied protein transport, protein folding, protein degradation, I had the great good fortune to go to WashU as a postdoc and work with Alan Schwartz, who's a member of the Institute of Medicine and just a fantastic mentor. And what I worked on was protein turnover, the ubiquitin system, and thoroughly enjoyed it. So I wanted to keep doing that, but because I lived permanently here in Baton Rouge, I decided to find a second postdoc where I could use what I had learned in something different. And so I came back and applied what I knew to adipocyte biology. And so, again, very nonlinear. But because of that, so I knew something about protein turnover, I knew something about adipocyte biology. It was a unique and novel field. And then I went to work at Pennington, and that's where I still am. So that's it for my career path. I think all I want to say to you is that if you think it has to be linear, it doesn't. You can be very successful and it not seem like it's going to happen at any given time. So what is the Ubiquitin-Proteasome System? I want to spend the rest of my time just talking about the kind of work we do in the lab. Okay. So at this point, this is a system that is really very well worked out. We know an awful lot about it. And this is essentially how it works. The substrates, and these are proteins in the cell, these are all intracellular proteins, are tagged by this polypeptide called ubiquitin. And I've shown that here in red. In order to do that, it requires this huge cascade of enzymes. And we know most of them, but there are probably a lot more that we haven't found out yet. There is a single E1 ubiquitin activating enzyme. So you have to think about that. In the cell, or system that's responsible for turning over almost all cellular proteins, there's only one enzyme that controls this system. So it really is a rate-determining step. There are a lot of enzymes that are called conjugating enzymes. These are the enzymes that when ubiquitin is bound to an E1, then these conjugating enzymes shuttle that ubiquitin to the final substrate. But there are potentially thousands of these E3s, ubiquitin ligases. In their job, their biochemistry is exactly what it says here. They are for ligating ubiquitin to a substrate. They're important here because this is the part of the system that determines specificity. So it's always the ligase that picks the substrate. So this system is what we call a very hierarchical system. There's one of this, maybe 100 or so of these, but potentially thousands of this. So at any rate, here's how it works. So you have a substrate. All these enzymes combine to add a ubiquitin to a substrate. And then it continues to add more and more. So you eventually have this whole, what we call ubiquitin ladder, a chain of these polypeptides that are bound to a substrate. And when that happens, let's back up here. So if you add one, this is a certain kind of signal. It is involved in endocytosis, it's involved in protein sorting, it's involved in gene expression, it's involved in cellular trafficking. If you add more than one in a very particular kind of configuration, so the cell really can sense exactly what the steric surface is when you add these polypeptides, then you get targeting for degradation at the proteasome, which is this huge protease complex in the cell. You get regulation of the cell cycle. You get, again, regulation of gene expression, of DNA repair, and of signal transduction. And so once these are added here, then they are removed and recycled. So at any given time in the cell, there is a lot of ubiquitin that's there to regulate the processes that are involved here. So I want to stop for a minute and give you a little bit of history of how this system works so you can get some idea of really how science progresses. Okay? And so we start in the 1930s here. All right? And so the thinking was in the 1930s, which is amazing to us now, that proteins are made and they are completely stable. They're only discarded just due to wear and tear. So everybody has the same stability. Nothing is different in the cell. And then in 1941, this researcher named Rudolf Scheinheimer used a stable isotope of nitrogen, which I'm sure you all are aware of, N15, which is used in some very basic experiments in biochemistry. And using this stable isotope, he was able to show that proteins are not stable, but they're in this dynamic state of being made and destroyed. And then they found that they were made and destroyed at very different rates. And so this was a complete paradigm shift in the 40s from what we understood in the 30s. In the mid-1950s, the lysosome was discovered. And so the thinking was that it's that the lysosome, where all of these proteins are degraded, and that is it. And so people thought, over, story is done. But in the 1950s and 60s, what was called the lysosomal hypothesis fell apart. It was found that it could not account for all this energy-dependent degradation that was going on of intracellular proteins. And so this really created quite a problem, and a lot of research was done trying to understand what was going on if it was not the lysosome. In 1964, this is one of the really important experiments in the field that I work, in the ubiquitin field. Rabinovich and Fisher took rabbit reticulocyte lysates, and they demonstrated that proteins can be degraded in them. And that was important because what they had learned from very basic experiments was that the reticulocytes no longer had lysosome. Those organelles were completely missing. And so now there was a system that could be manipulated and modeled where you could look and ask the question, so what is the protease that's responsible for degrading all of these proteins that the lysosome is not involved in? And this system is what allowed people to understand the Ubiquitin-Proteasome System. In 1977, Ettlinger and Goldberg, and Fred Goldberg is still one of the leading researchers in this field. He is an amazing researcher. What they did was they took the reticulocyte lysates and they isolated from it the protease that was responsible for degrading all the proteins in the reticulocytes. And many years later, this was defined as the 26S proteasome. And the 26S refers to how it sediments, and it sediments is a huge, huge complex. And in fact, this is a protease that you can see on EM. It is really, really beautiful. If you get a chance to go and just Google it or look it up in PubMed and see some of the EMs of this structure, it's really very nice. 1978, this was another seminal experiment. Conover, Hod, and Hershko discovered in this lysate that there was a polypeptide that was stable to heat, and that this was absolutely required for the protein degradation in these lysates, and they called it APF1. But this APF1 is a polypeptide that we now know as ubiquitin. This was a paper that was published in a journal called BBRC, and this is another lesson I think that's really important. And BBRC is not considered to be a good journal at all. It was not considered to be a good journal then, and it isn't now. But it's very important. This paper was the most seminal paper in this entire field, and it was published there because it was so novel and so different that other journals would not accept it. And Conover and Hershko in 2000 won the Nobel Prize based primarily on what they did in this paper published in a journal that's not considered to be all that important. So you do your work, and you think it's important, and you get it published, and you never know what's going to happen. So in the 1980s and the 1990s, not that long ago really, all the enzymes of the ubiquitin system have been identified, or were identified. So the whole hierarchical system, the E1, their E2s, and the E3s were identified. There's also potentially E4, so the story's not quite over yet. In 1986, again, not long ago at all in terms of scientific discovery, this proteasome, the 26 proteasome, was identified as the protease responsible for degrading ubiquitin-modified proteins. And again, this was in reticulocyte lysates. In between 1998 and 2000, we were able to show that ubiquitin-proteasome-dependent degradation occurs in the nucleus. So this happened when I was in the lab with Alan Schwartz, and Aaron Jaconover was also in the lab at the time. And this was important. Another group had shown it in yeast, and we showed it in mammalian systems. It's important because now we know that the ubiquitin system is intimately involved in regulating transcription, in regulating DNA repair, all sorts of things that go on nuclear function. And it doesn't require moving anything out of the cytoplasm to do it. So this system is part and parcel of chromatin. And here we are now, where we really are very clear that ubiquitin-proteasome system is involved in everything the cell does. It's a highly regulated system. So degradation of proteins is not an afterthought at all. It's a regulatory system. It's part of signal transduction. And my bias is that I would like for you, whatever you're doing, give it a thought. Does it have anything to do with what you're looking at? It probably does. So this is just an example of how we look and say, is something important? We look at how many publications are out on the topic. And what you can see here, in 1980, so if you remember, I told you not Okay, is that working? Yeah. Okay, so in 1980, we didn't know a whole lot, right? So, but then, a little bit after that, in 86, we learned that ubiquitin conjugation occurs to substrates. We go on here, and then not a lot of publications. We go here, the proteasome was discovered. This is 86. In 1990, the really interesting thing was someone synthesized in a chemistry lab an inhibitor of the proteasome. And that allowed people to manipulate the system, and research really began to take off. Here, in the late 90s, a drug called bortezomib was made, and this is clinically relevant because this is a proteasome inhibitor that's used to treat multiple myeloma. So this is a system that went from being unknown in 1990 and 1986 to a clinical compound before 2000. So that's really moving along very, very quickly. You can see here cumulatively the papers now are almost 40,000 on the topic. And here there's now an inhibitor for the activating enzyme. So I think things will really pick up even more as we learn how to manipulate biochemistry of the system. So just again to reiterate this, that the ubiquitin system is involved in every aspect of the cell, whether it's insulin signaling, which is what we're interested in primarily, lipid metabolism, amino acid metabolism, cell signal transduction, quality control, protein folding, endocytosis, the response to hypoxia, cell cycles, circadian rhythms, transcription, chromatin modeling, epigenetics, DNA repair, and nuclear transport. And if you think about diseases that are associated with each of these things, then you have neurodegenerative diseases, you have genetic diseases like Angelman syndrome, Little disease, cystic fibrosis. Here, all the cancers that are involved in cell cycle control. Here, diabetes, obesity, and muscle wasting are all involved with the ubiquitin system and insulin signaling. So here's what we're interested in. We want to understand the role of the ubiquitin-proteasome system and the adipocyte biology. And here, these are all, it's one of my favorite pictures of fat cells. All of these phase bright structures here are lipid droplets that are in adipocytes in culture. And this is what we use a lot of. So when we want to look at this system in adipocytes, where do you start doing this? And you always have this problem in experiments. Where do you start? And the place that we started was picking a substrate that we could tease it apart and see what goes on. And that substrate is PPARgamma in adipocytes because this is a transcription factor that is absolutely required for forming a fat cell. If you don't have it, you don't get a fat cell. It doesn't matter what else is in the system. You won't get one. But if you have it, you don't really have to add a whole lot else. You can get a fat cell from a skin surface fibroblast. It's that critical. So we thought if we can just start understanding this protein and how it's regulated by the ubiquitin system, then we'll understand something about adipocyte biology. So one of our first experiments, and this has been quite a while back, was to ask the question, if we activate this protein, do we change its stability? And so the way that the protein is activated is with a compound called TZDs. And these are actually the compound that's the drug that's used to treat diabetes. It's now going off the market because it's a little dangerous, it seems. But if you have relatives who have type 2 diabetes, they probably have taken TZDs. They're called Avandia. So look around at home and ask yourself, do you know anyone who's ever taken Avandia? If they have, then this is how it's acting. It's acting on PPARgamma. But here's what we found. So we look at turnover rate. So this is the time from the time the protein is synthesized until it's destroyed in the cell. And there are two forms of PPARgamma. So there's gamma 1 and gamma 2 in adipocytes. Gamma 2 is around for about an hour and a half under basal control conditions. If you add this ligand, which directly binds PPARgamma, then the time that it hangs around in the cell is reduced to an hour. But if you add an inhibitor of the proteasome, then no matter what you do, whether it's basal or the ligand is there, then the time it stays in the cell is dramatically increased. It's 13 hours. And this is true as well for PPARgamma 1. So it's a little bit less stable than gamma 2. It hangs around for almost an hour. It decreases to a little over 30 minutes, 40 minutes. But it hangs around for 10 hours if you inhibit the proteasome. So this was our first experiment that showed that PPARgamma in fat cells is regulated by the proteasome, and that its activation is related to the proteasome. So just a summary of what we've learned from earlier studies that have gone on over the years. So this is a typical drawing you'll see of proteins, how they look. So this is PPARgamma. It's 507 amino acids. Gamma 2 starts here. Gamma 1 starts here. The only difference in these two forms in the same tissue is 30 amino acids at the front of the N-terminus of it. So there's a domain here we call the AF1 domain. Here's the DNA binding domain, the hinge domain, and the ligand binding domain. And here's an activation domain. And so what we've learned over time, that I've already told you, that they're degraded by the proteasome, that there's proteasomal degradation of PPAR that's ubiquitin independent, that goes on, involves this domain somehow, and ubiquitin dependent, which involves this domain. We know, using the inhibitors of E1s, that a functional ubiquitin system is absolutely required to activate this protein in fat, and that's extremely important. It says to us, and we've learned in other experiments, that before you can activate a protein that's this important to a cell, that the cell ensures it's going to get rid of it, that it won't stay activated for too long. So it's really a very elegant system. And we know that PPARgamma is modified by ubiquitin, as shown here in the ligand binding domain, but we don't know exactly what residue yet. But we do know that this domain here, the AF2 domain, overlaps the domain that's recognized by the ubiquitin system. So it is related to activation, as we would have predicted by the fact that a functional system is required for its activation. So then we wanted to know, okay, how exactly does PPARgamma interact with this ubiquitin system? And we think of it, although we don't know this is the case, but we think of it as being bound to DNA when it interacts with it. So the question is, we have an E1, an E2, and an E3 that we are assuming is interacting with PPARgamma. And so this is where we focused. We wanted to identify an E3 ligase, the ubiquitin system, that's directly affecting PPARgamma stability, activity, and ubiquitination. And so how do we go about doing this kind of thing? So what we did was we used RNA interference to inhibit gene expression of a number of ubiquitin ligases. In fact, we screened over 250 of them in an in vitro system where we grew fat cells, and we can do this and induce them to 100%. So you can see this here. We use OLED-O staining of neutral lipids, so this binds to neutral lipids. Fat cells obviously have a lot of that, and you'll see red wherever there's lipids in the fat. If you look at the bright field, it may be a little hard to see, but there are phase bright structures here. Those are lipid droplets in the fat cells. So this was our model system here, and then what we did was we did a screening of a library, again, over 250 individual siRNAs that we inserted into these cells. And then we ask ourselves exactly what happened to the protein levels of PPARgamma. Now, here's how we know that we got the ligase of the siRNA targeting the ligases into the cell, is that it's attached to a fluorescent tag. And so wherever this reagent is in the cell, it will fluoresce red. This is what you see here. If you blow up this little section here, you can see that we have it almost in every cell here. So it's a system that worked very well for us. And then if we look at the fluorescent tag for the siRNA itself, and we ask, is this in the nucleus or is it in the cytoplasm? And what we can see is that it's in the cytoplasm. We stain the nucleus blue here with a DAPI stain. This is important because this RNAi system works in the cytoplasm. It doesn't work in the nucleus. So we're talking about binding to the message of the RNA and destroying it. So that's how you regulate the gene expression. It's not binding to DNA. And so this was our system, and it worked really well for us. This is an example of how we screened these ligases. So we ran Western blots, and we looked at protein levels, since this is what we're interested in. So here, there are these two bands of PPAR. This is the gamma-2 and the gamma-1. These are basal conditions, so every gel has to have a set of controls here. This is what the levels look like under controlled conditions. This is when we add the TZDs, the levels go down. This is consistent with it being targeted for degradation by the proteasome. If we have MG132, then you can see the levels go up. So there's more of it there. So these are our controls. We're looking for something that's comparable to this when we ask if we get rid of the ligase, do we stabilize this protein as if it's never recognized by the proteasome. So it's the same as the proteasome inhibitor. So this was an siRNA that didn't target anything. It was directed to luciferase, which is not found in mammalian cells. These are all numbered. We didn't know what they were at the time. So this is negative. This looked interesting. Negative, negative, interesting, interesting, negative, maybe interesting. We didn't really pursue this. But we picked this one here, 3B07, and that's SIA2. And the reason that we picked it to start with, although there are others that are interesting, is that something was known about this protein. It was known using yeast 2 hybrid systems that it affected the degradation of a protein called NCOR, which is a co-repressor of PPARgamma. And so we thought, okay, let's start here. We have a little bit of a lead into this system. So this is what SIA2 looks like. It has a ring finger at the N-terminus that is actually doesn't mean anything other than really interesting new gene, is how it was named when it was discovered, this domain in proteins. It has zinc fingers. What this means is these are DNA binding domains. They have a lot of cysteines and histidines, and they bind zinc, which holds that structure together. And then it has a domain at the C-terminus, which is for homodimerization of SIA2 itself and for substrate binding. So the E2s bind this ligase here at the N-terminus, and then as bound to this ligase, they shuttle the ubiquitin off to a substrate as this process goes on and on and on until it's recognized by the proteasome. So what is known about this protein, this was found in Drosophila originally, so in the fruit fly. What was known is it's involved in RAS signaling, and it's involved in epidermal growth factor signaling, and this is in photoreception in the Drosophila. And so you have this receptor tyrosine kinase, EGFR, that signals by the RAS pathway, you probably know a good bit about that, to the nucleus where SIA2 is found, and here it regulates ubiquitination of a substrate, target gene expression, and proliferation. So that's what was known in Drosophila. So I'm not going to go over the experiments for what we've done to really look at this in the past, but just to summarize them here, again with this structure of PPAR, what we know now is that SIA2 regulates PPARgamma at steady-state protein levels. We know that it has nothing to do with regulating PPARgamma gene expression. So its effect on PPAR is post-transcriptional. We think it's post-translational, but we know that it's post-transcriptional. We know that it regulates ubiquitin modification of PPARgamma. And now we know that its expression is upregulated with the formation of fat. And I should back up here and say that although we know it regulates ubiquitin modification of PPAR, We don't know if it directly binds PPARgamma. In fact, we suspect it doesn't, because when you look at the structure of PPAR, it doesn't have the domain that SIO2 recognizes. So we think that what we found is an indirect regulator. There's probably another ligase that we'll have to look for eventually to really understand this system. But right now, we're focusing quite a bit on how it's upregulated in adipogenesis. And so we ask the question, does it play a role in forming fat cells? And the reason we think that's reasonable is because it's upregulated when you form fat cells, and we know that it affects PPARgamma stability and activity, I should say. And we know that PPARgamma is absolutely essential for forming fat cells. So this is our rationale for asking this question. And so we do some really straightforward kind of experiments. I won't go into all of them that we've done. But the thing you have to do first is say, if you don't have this protein there, can you get a fat cell formed in cells that are really pre-adipocytes? They're primed and ready to form a fat cell. And this is what happens. So it's the 3T3L1 model system that we use. These are the pre-adipocytes. They will form fat cells under the right conditions. This is a bright field image. You can see we've got these phase bright lipid droplets. Here, this is GF. Okay. All right, so this is our control here. So we can get all red O staining of the 3T3L1. Here we do these very stable cell lines where we introduce the shRNA that's targeting luciferase. So this is a negative control. We shouldn't see any effect here. And you see the phase bright here. This is lipid droplets. But we have the shRNA in there because we get the GFP signal. The green fluorescent protein, so this is our control for the experiment working. Plenty of fat because the OREDO is there. Now, if we delete SIA2 by using shRNA to SIA2, you see in the same conditions, you don't see phase bright cells here at all. We still have the GFP there, and we don't have the OREDO to the same extent. So we just don't get whatever this process is that's required to form fat cells. So this experiment says that SIA2 is essential for forming fat. Well, that's nice. So you lose it and you don't get it. But what happens if you overexpress this protein? Can you make fat in a cell type which has no intention of becoming fat? So this is what we see. So now we overexpress SIA2 using different technology in fibroblasts. So these are like cells from your skin surface. And here's what we see. So we use a retroviral vector called pBAY. So this is the vector alone. There's nothing attached to it. And you can see without the ligand, you don't see any staining with the OREDO. Even with the ligand, you only see a little bit of staining. So even if we just put the vector in, we get a little bit of response if we have the activator of PPAR here. If we put in this vector overexpressing SIA2, so this is a lot of this protein that's now being expressed in these fibroblasts. This is what we see with no vector, with no ligand, sorry. We don't get anything. With ligand, we see OREDO staining. So this protein alone, with the ligand there, is sufficient to form fat cells, according to the OREDO staining. And our positive control here is if we overexpress PPARgamma in this same cell type. So we know that it can cause adipocyte formation. So without the ligand, this is what we see, but with it, we get a lot of fat. So we learned two things here, that if we overexpress SIA2, that's sufficient to form fat cells, but it doesn't quite reach the level of adding PPAR itself. So it's not all that's required, and that should be obvious, actually. So this is the last data slide I'm going to show you. So now we started to do work with animals which have this protein knocked out in them. So this is genetic manipulation of the animals so that they don't express SIA2. They don't express SIA2 in any of their tissue at all. So this is called a global knockout of the gene. And this is what we see in some of their adipose tissue. So remember, I'm saying that SIA2 is required to form fat. So we thought these animals, no matter what we did to them on a high-fat diet or not, they would stay lean, and they would have some trouble with being lean. But they didn't stay lean. And this is another thing that you'll learn doing research, is that you will have a hypothesis, and it's generally going to be wrong. And ours was wrong here, but very interesting. So they got just as obese as the wild-type animal when we put them on a high-fat diet. But there is something different about them. So if we look at the wild-type animal and we look at the adipocytes from one of their adipose tissue depots, here's what we see. We see a lot of cells, and they're relatively small. If you look at this same depot from our animals where we don't have SIA2, then there are far fewer cells, and they are large. So there is a difference in vivo, which makes sense really compared to our in vitro findings that it's required for adipogenesis, but not the only factor that's important. So we don't know whether we have something that's compensating here or whether, we just SIA2 in and of itself is not quite enough. But this is consistent, we think, with what we see in vitro, that it's important for forming fat cells. It's also interesting because there are a lot of models of animals out there now that say the larger the fat cell you have, the more insulin resistant you will be. So the size of the fat cell in your body is really very important. But our animals are more insulin sensitive than the wild type animals, although they are just as obese and have very large fat cells. And there are a couple of other labs that have results that are a little bit like this now. So there's this emerging idea that it's not necessarily true that small fat cells mean that you will be more insulin sensitive. So we have a lot of work to do here to understand what's going on, but we have a very good start at this. So what's next for us in the lab? So we're doing a couple of things now. We're very interested in understanding how does SIA2 affect PPARgamma using the mouse model. And so we have a lot of tissue we have to look at, a lot of things we have to assay with all of the tissue. This is important clinically because when you activate PPARgamma in fat, a reciprocal thing happens in bone. You lose bone. So if you've had relatives who are on Advandia, and particularly if they're older, then what may be happening to them is while they're on this and it helps with their blood glucose levels, their bones are not as healthy as they were before they were on this drug. Because the more PPARgamma you have, the more you'll form fat within the bone marrow itself, and it makes the bone more fragile. And so there's people who've had bone fractures. We think, based on our studies now, that SIA2 is playing a role far upstream in deciding whether in stem cells, whether they become fat or bone. And so this could have some important implications for the treatments of type 2 diabetes. So this is where we are now. I just want to acknowledge the people who are doing most of this work in my lab. Gail Kilroy is the research associate, and Heather Kirk-Ballard, who just got her PhD. Lauren Carter is here. She's the HHMI student, and she's doing some really interesting work on the physiology of how SIA2 is affecting one of the endocrine hormones called FGF21 and its role in forming fat and bone. And then we've had Tanya Acharya in the lab a couple of years ago who worked on a different project. These are the people who help us with our animal studies, Barry and Cindy and Tamara Mendoza. This is a group from another group at Pennington who we collaborate with on skeletal muscle studies, along with David Rudnicki at Rutgers. Someone at Stanford and Peter McCallum. Stanford's in California, Peter McCallum in Australia. These were their mice that they agreed to share with us. We do work with Steve Smith at Sanford Burnham in Florida on a different aspect of this study. And we've gotten a lot of advice from Ormond McDougall at the University of Michigan and Will Cawthorn, who works in his lab at the University of Michigan. I add this just to show you that science is really a highly collaborative endeavor. So you'll never be able to do it just by walling yourself off and thinking about things all by yourself. You're going to need a lot of help from a lot of different people to be successful in it. And the work in our lab is funded by the American Diabetes Association and the NIH as at the NIDDK and with a commercial endeavor in conjunction with New Potential here in town. So that's it. Do you have any questions? They, when we started the study, again, we predicted that they would not gain as much weight. In particular, they wouldn't gain as much fat. But they gain weight at the same rate, and they gain fat at the same rate on a high-fat diet. You know, we can't quite put our finger on it. There's something about them that still seems a little different, whether the depot size, just to the eye, whether the fat depot size is a little larger, whether their lean body mass may be slightly smaller. It's just not quite clear to us. But the simple answer is they get just as obese as the wild-type animals. But the important thing is that even though they do that, they're very insulin sensitive. They are as insulin sensitive when we measure it by doing insulin tolerance test and glucose tolerance test as the wild-type mice on a lean diet, on a low-fat diet. So even though they're obese, it's not causing them problems with glucose disposal. Let's all thank Dr. Floyd for being with us this morning.